MF approach, or membrane filtration approach, to test for fecal coliforms in a water sample. And you can use this approach to test for any kind of bacteria in a water sample, or any fluid for that matter. You could use it for apple juice. And so the water is collected. Typically, you start with a sterilized little plastic collection bottle. And then the water sample can be brought to the lab, where it's run through a membrane filter apparatus like you see here. And so the basic workflow looks like this. You have your water sample. The water is passed through the filter. You use some sterile tweezers to pull the filter off and apply it to a selective and differential medium for the detection of whatever you're looking for. In this example, we're looking for fecal bacteria, or evidence of fecal bacteria in the water. Did something poop in that stream, for example, that woman was taking a sample from? In this cartoon, you're basically seeing the difference between total coliforms, which are a whole group of gram-negative rods with certain characteristics, and then fecal coliforms, which are more specifically associated with the gut tract of animals. And so if this animal poops into the environment, those fecal coliforms will mix with the total coliforms. And so we can actually use different kinds of media to distinguish between just background coliforms that are normally present in the environment, and fecal bacteria, which are not normally present in the environment, and therefore indicate the possibility that more contamination is present. So here are those bacteria growing exponentially in the environment. And so one of the plates that you might use in the filtration approach will allow you to distinguish between E. coli and other coliforms. Hardy Diagnostics in Santa Maria, which we work closely with, has a chromogenic type medium like this that allows you to distinguish between E. coli and other coliforms. And so the E. coli cells will form very large bluish colonies on this particular medium, whereas other coliforms will be smaller and red. So imagine I filtered 100 milliliters of water through the filter apparatus. I can then count 1, 2, 3, 4, 5, 6, 7 fecal coliforms per 100 milliliters of water. And then I can use that information to determine whether or not the water is drinkable or even safe for recreation. And you can find standards like that in the FDA website and so forth. So for example, any water sample that contains any detectable E. coli is not considered safe for consumption. But there's different standards, obviously, to swim in that water. Another thing that we can do, though, is incubate plates at a slightly higher temperature. So in this particular case, 44.5. The previous plate was held at 35. So all coliforms seem to grow equally well at that temperature, but when you raise the temperature to 44.5, now you can specifically target the more heat-resistant fecal coliforms. And so that's another way to get a more specific count without the background total coliforms. But sometimes you want total coliforms. So here's our setup, and you see a vacuum pump, Erlenmeyer flask, a sterilized filter apparatus, and then you're going to apply a sterile filter. Usually it's a 0.2 micron filter, so the pore sizes are about 0.2 microns. And a typical bacterium like E. coli is around 3 microns long and maybe 1 to a half micron in diameter, so it shouldn't go through the pores. So in this section of the video, the individual's applying the sterile filter to the sterilized filter apparatus. And now we're ready to apply a water sample to that. And so the water sample is applied. And then the water sample is pulled through. See it going down. And then the individual can pull using sterile forceps or tweezers, pull the filter off and apply it to a selective or differential medium for the growth of coliforms. And then we put the plate in the incubator.